Hi, greetings of the day. Today we shall start discussing proteins and their biosynthesis. This is the first lecture in this series. As you already know that the genes in DNA encode protein molecules, which are the workhorses of the cell, carrying out all the functions necessary for life. For example, enzymes encoding those that metabolize nutrients and synthesize new cellular constituents, as well as DNA polymerases and other enzymes that make copies of DNA during cell division, or all proteins. So these proteins are the main determinants of biological form and function. Therefore, they heavily influence the shape, color, size, behavior, and physiology of organisms. So, because genes function by encoding proteins, understanding the nature of proteins is essential to understand gene function. So, if we recall, in the simplest sense, the expression of a gene means manufacturing its corresponding protein, and this multi-layered process has two major steps. In the first step, the information in DNA, as we will see here, so the information in DNA is transferred to messenger RNA molecules by way of a process called transcription that we discussed earlier. So during transcription, the DNA of gene serves as a template for complementary base pairing, and an enzyme called RNA polymerase 2, it catalyzes the formation of pre-mRNA molecule, which is then processed to form mature RNA. It's here. So later on, the resulting mRNA is a single standard copy of the gene, which next must be translated into protein molecule. So a protein is a polymer which is composed of monomers called amino acids. In other words, a protein is a chain of amino acids. So because amino acids were once called peptides, the chain is sometimes referred to as a polypeptide. Amino acids have the general formula, as you can see here. All amino acids have a side chain or R reactive group, which is here. And as you know that there are 20 amino acids known to exist in proteins, each having a different R group that gives the amino acid its unique properties. The side chain can be anything from a hydrogen atom, as it is in glycine, to a complex ring, as in amino acid tryptophan, as it's here. So here, the structures of other amino acids, you can see there are 20 different structures. I hope you have already memorized the names and structures of amino acids in your chemistry or even in biochemistry classes. So in protein, the amino acids are linked together by covalent bonds called peptide bonds. A peptide bond is formed by the linkage of carboxyl end, which is here, of one amino acid with the amino end of another amino acid, which is this end. Each amino acid, which is double A, which or each AA indicates an amino acid, and R1, R2, R3 represents R groups or side chains, that differentiate the amino acids. So one water molecule is removed during this reaction, and because of the way in which the peptide bond forms, a polypeptide chain always has an amino end, this one, and and carboxyl end, which is at this end. So there are four levels of protein structure that are distinguished from one another by the degree of complexity in the polypeptide chain. A single protein molecule may contain one or more of protein structure types. These structure types are primary, secondary, tertiary, and quaternary structures. So we shall discuss them one by one. So this is the primary structure of a protein. The primary structure describes the unique order in which amino acids are linked together to form a protein. The amino acid sequence of a protein is determined by the information found in the cellular genetic code. The genetic code will be our next topic. The order of amino acids in a polypeptide chain is unique and specific to a particular protein. Altering a single amino acid may or may not affect the function of the protein. So this is the secondary structure of protein, where the local regions of the polypeptide chain fold into specific shapes called the protein stringent structures. In other words, it refers to the coiling or folding of a polypeptide chain that gives the protein its 3D shape. So each shape arises from the bonding of forces between amino acids that are close together in the linear sequence, as you can see here. So these are two different amino acids, and the hydrogen bonds between these amino acids different equations give uh, give a particular structure to the protein. See, the most common secondary structures are alpha helix, this one, and the pleated or beta sheets, which is here. Different proteins show either one or the other, or sometimes both of these structures. Uh, the pleated sheet has two polypeptide segments, which are arranged in opposite polarity as indicated by arrows. So the tertiary structure is produced by the folding of secondary structure. There are several types of bonds and forces that hold a protein in its tertiary structure. These include hydrophobic interactions that greatly contribute to the folding and shape of a protein. The R group of the amino acid is either hydrophobic or hydrophilic. The amino acids with hydrophilic R groups will seek contact with their aqueous environment, while amino acids with the hydrophobic R groups will seek to avoid water and position themselves towards the center of protein. Similarly, the hydrogen bonding in the polypeptide chain and between amino acid R groups helps to stabilize protein structure by holding the protein in the shape established by the hydrophobic interactions. So, due to protein folding, ionic bonding can occur between the positively and negatively charged R groups that come in close contact with one another. Folding can also result in covalent bonding between the R groups of cysteine amino acids. So this type of bond forms and it is called a disulfide bridge. Interactions called van der Waals forces also assist in the stabilization of protein structure. So these interactions pertain to the attractive and repulsive forces that occur between molecules that become polarized. So these forces contribute to the bonding that occurs between molecules. Some proteins have quaternary structures. Such a protein is composed of two or more separate folded polypeptides, also called subunits. These subunits are joined by weak bonds. The quaternary association can be between different types of polypeptides, resulting in a heterodimer if there are two subunits, or between identical polypeptides making a homodimer. So hemoglobin, which is an example of heterotetramer, where there are four different subunits from two different proteins. Uh, so it is composed of two copies, each of two different polypeptides, which is shown in green and in purple here. So many proteins are compact in structure. I mean, there are other proteins uh, which are compact and they are called globular proteins. There are enzymes and antibodies that are among the best known examples of globular proteins. Proteins with linear shape are called fibrous proteins. They are also important components of substructures such as skin, hair, and tendons. So let's compare the primary, secondary, and tertiary structures. So we can say that primary structure is a sequence of amino acids, simply a linear sequence of amino acids. Secondary structure is the folding of peptide chains into an alpha helix or beta sheets. There are two different types. Tertiary structure is a three-dimensional structure of protein. Similarly, primary structure is linear. Secondary structures are either in the form of alpha helix or beta sheets, and tertiary structure is in globular form. Primary structure is composed of peptide bonds formed between amino acids. Secondary structure encompasses hydrogen bonds, and tertiary structure involves disulfide bridges, salt bridges, hydrogen bonds, and other form of bondings. Primary structure is formed during translation, while strenic structure forms collagen, elastin, actin, myosin, and keratin-like fibers. Similarly, tertiary structure is included in proteins, in hormones, in albumins, globulins, and even in hemoglobin. Primary structure is involved in translational, post-translational modifications. Similarly, secondary structure is involved in forming structures such as cartilages, ligaments, skin, and tertiary structure is involved in metabolic functions of the body. So, if we conclude, for proteins, shape is all important for a protein because a protein's specific shape enables it to do its specific job in the cell. A protein's shape is determined by its primary amino acid sequence and by conditions in the cell that promote the folding and bonding necessary to form a higher level structure. The folding of proteins into their correct conformation will be discussed later on, and the amino acid sequence also determines which R groups are present at specific positions and thus available to bind with other cellular components. The active sites, for example, here we will discuss the enzymes. The active sites of enzymes are good examples of precise interaction of R groups. Each enzyme has a pocket called the active sites into which the substrates or other substances uh, can fit. So within these active sites, the R groups of chain amino acids are strategically positioned to interact with a substrate and catalyze a specific chemical reaction. Similarly, there are domains of the proteins that are distinct functional and structural units in a protein. So usually they are responsible for a particular function or interaction contributing to overall role of a protein. Domains may exist in a variety of biological contexts where similar domains can be found in proteins with different functions. For example, SRC homology 3 domains are small domains of around 50 amino acids that are involved in protein-protein interaction. So SH3 domains have a characteristic 3D structure. They occur in a diverse range of proteins with different functions, including adapter proteins, phosphatidylinositol kinases, 3 kinases, phospholipases, myosins. There are some proteins which include only one SH3 domains, and there are other proteins which contain multiple SH3 domains. So how this shape of the protein is changed. So if the protein is subjected to changes in temperature, pH, or it is exposed to chemicals, the internal interactions between proteins, amino acids can be altered, which in turn may which in turn may alter the shape of protein. Although the amino acid sequence, also known as protein primary structure, does not change, the protein's shape may change so that it becomes dysfunctional, in which case the protein is considered denatured. For example, pepsin is the enzyme that breaks down protein in the stomach. So this enzyme only operates at a very low pH. At higher pH, pepsin's conformation, the way its polypeptide chain is folded up in 3 dimensions, begins to change. The stomach maintains a very low pH to ensure that pepsin continues to digest protein and does not denature. So there are some other examples like chaperone proteins, which help some proteins fold into correct shape, and therefore they help in maintaining the function of those proteins. So here are some examples of the proteins and their functions, how they are involved in different functions in this in the body. So there are several types of proteins, for example, enzymes, transport proteins, structural proteins, and they perform a variety of functions. Like enzymes help in food digestion. Insulin coordinates the activity of different body systems. Hormones, for example, including insulin, yes. And there are defense proteins which protect the body from foreign pathogens. There are storage proteins, especially in plants, for example, or even in animals, like egg proteins. They provide nourishment in early development of embryo and the seedling of plants. So there are a lot of other examples. We have discussed just few of them. If we summarize our lecture, protein structure depends on its amino acid sequence and local low energy chemical bonds between atoms in both the polypeptide backbone and in amino acid side chains. Protein structure plays a key role in its function. So if a protein loses its shape at any structural level, it may no longer be functional. Primary structure is the amino acid sequence. Secondary structure is local interactions between stretches of a polypeptide chain and includes alpha helix and beta pleated sheet structures. Tertiary structure is the overall, the three-dimension folding driven largely by interaction between R groups. And quaternary structure is the orientation and arrangement of subunits in a multi-subunit protein. I think that's all for this topic. And the next topic will be the genetic code. Thank you.